That sounds better. Thank you, Mr. Chairman, and thank you, Director Zhang and the organising committee for inviting me here today. I was at the Institute Pasteur 3 years ago, and it's my distinct pleasure to be back here again. When I was here in 2022, I met Regis Grail, and we've worked together ever since, so it's been a good collaboration. And a special honour for me to be the first speaker. I think it's because I'm the oldest person in the room, so thank you for that as well. So we talk about influenza and how does that fit with an unmet need? Well, I hope I'll show you in the next 25 minutes that there's still a lot of work to do to get effective, long-lasting influenza vaccines for both seasonal and future pandemics. So I'm going to go through a few different topics here. I won't dwell on this. Okay, so this is, okay, we've got a little trouble with our translation here. So these was meant to show the influenza surveillance network from WHO. The only thing it's missing is the map, but what we have there in the red is the influenza centres that are called collaborating centres. So they are the top of a very large WHO network of over 150 national influenza centres. And there's a National Influenza Centre, of course, in here in Korea. And the ones in blue are centres which focus on the interplay between animal and human influenza viruses, which is very important in this time. And just to show you where we are in Melbourne, and all the pictures have gone, so this is going to be a fun presentation. But we're in Melbourne, which is in the southern part of Australia, and we're very close to the reference laboratory in Geelong. And we have a number of laboratories there. We're a laboratory-based organisation, and we have some specialised laboratories for doing some vaccine seed development, and we have animal facilities for BSL-2 work, as well as high containment areas for BSL-3 work with highly pathogenic avian influenza viruses. Okay, all right, this is not looking good, I'm afraid, people. I should have checked these before we came up. But let's just talk about influenza. So influenza comes in 4 different varieties, A, B, C, and D. That's the simple part. So influenza A has 18 different subtypes. H1, this is the hemagglutinin terminology, and goes through to H18. So a lot of different types of influenza in the group A, and they have a very wide host range. So they can infect wild birds, they can infect a whole range of different animals. And we've seen recently in the United States, even dairy cows can become infected with the H5 variety of influenza virus. However, there have only been 3 different subtypes which have infected humans, and that's been H1, H2, and H3. For influenza B, there's 2 different varieties, B Victoria and B Yamagata. Fortunately, the B Yamagata is now extinct. And for type C, we have only one type of influenza C, and that affects mainly children. And we don't have any vaccines for influenza C, but it does have some impact, especially in children. Type D mainly infects cattle at this stage. Okay, we've got a slide with everything on it. We're going better. So this is what happens in the world of influenza. We have pandemics, and then sometimes these viruses stay around and sometimes they die out when the next pandemic comes along. So you can see here we've had a number of pandemics over the last 100 years shown in the starbursts there, and currently we're still carrying the H3N2 from 1968, which is currently circulating, and the H1 changed over in 2009, if you can remember back that far, when there was a pandemic. And for the influenza B viruses, these have continued right through this period. However, there was a split back in the 1970s into these 2 distinct lineages, the B Yamagata and the B Victoria lineage. And interestingly, the B Yamagata lineage is now extinct, as I'll show you. So we're based at the Doherty Institute. That's the building in the middle there. Some pictures of our labs, and the Geelong veterinary facility, which is the Australian reference laboratory for veterinary infectious diseases. Okay, and there's some nice pictures too, so that's what you missed out on. Lots of different animals. So I should have just mentioned wild birds are the main reservoir for influenza A. Okay, we've been through that. Seasonal influenzas have been around since the 1950s, first produced in 1947 by Sanofi here, shown in this diagram. And we've gone from bivalent vaccines to trivalent vaccines to quadrivalent vaccines, and now we're moving back to trivalent vaccines. Why are we doing that? Well, this comes from a recommendation. I think we might have skipped a few slides here, but it doesn't really matter. So this, we first noticed that the B. yamagata viruses were dropping out, and so WHO made a recommendation back in September in 2023 that because these viruses had died out after the pandemic, that we would no longer need that component in the vaccine. So now we're back to a trivalent vaccine, and by the time next year comes around, nearly all seasonal influenza vaccines will have gone back to a trivalent vaccine after about 10 years of making quadrivalent vaccines, where we had 2 influenza B viruses in the vaccine. Okay, so the B. yamagata is no longer circulating, so that is now going to be removed from the vaccine. What does that give us in terms of an opportunity to do something different with our standard vaccines? We could add 2 H3 components. We could add 2 H1 components, or we could add more of one component. We could have double H3 or double H1. So what makes sense? And what makes sense from our perspective is the component, which is the H3N2, which has been around since 1968, usually has the poorest vaccine effectiveness when we look at these meta-analyses. So this is shown here in this graphic here. This is a pooled meta-analysis looking at the vaccine effectiveness for H3, H1, and B viruses. And you can see that H3s generally are the lowest effectiveness in the community. So if we're going to try and improve something, it should be the H3 component. So we'll cross out the H1 and we'll focus on 2 H3s, or potentially we could still add more of one component now that the B. yamagata lineage has been removed. So this is a situation in which we held, we're part of the WHO group that makes the recommendations for influenza vaccines. And this is a situation we faced in a very short time ago, that human H3 viruses were showing genetic heterogeneity. There were a number of genetically different clades that were showing convergent evolution. There was a small outbreak at the end of the season in one country with a variant H3 virus that had a number of changes which we know are antigenically different. But the majority of the viruses which are still in circulation didn't have these changes. So what should we do? Should we just go with the majority of the viruses, or should we select a variant which has the potential to evade the vaccine if it emerges and spreads? And you've got to remember, when we're making these decisions, there's 9 months gap between when we make that decision and when these viruses are likely to circulate. So why not pick both? Well, we can't, because there's no regulatory authority to do that. So we need to build some data to support those sorts of changes. So we went into collaboration with Sanofi, and Sanofi have been quite interested in this proposal. So instead of having one H3 component, we had 2 H3 components. So we took the 2023-2024 Northern Hemisphere formulation and added an extra component, the A-Tasmania 503, which was from 2021 recommendation. So you can see there that this was a fairly small study, 50 people in each group, and we looked at the straight trivalent vaccine, a double dose of the Darwin, and addition of the Tasmania to the trivalent vaccine. So this was done in 18 to 49-year-olds firstly, and then in greater than 60-year-olds to see whether this same approach would be beneficial not only for adults but for older adults. So what we're trying to do is broaden the coverage of these vaccines, or increase the magnitude of these responses to this vaccine. You'll probably know that Sanofi have a high-dose vaccine which has 4 times the amount of hemagglutinin. This would only have double the amount of hemagglutinin. So would that have any impact? And can we get a broader response? So taking strain A and looking at trying to get coverage across all of these other virus strains which are circulating and combining them to give a broader coverage. Unfortunately, I can't show you any data, but the analysis is ongoing. What has happened is that Moderna have done these studies. They've done them more than a year ago, and they've reported them in the literature. This is using mRNA. So adding things to mRNA is much easier. So they've added 1 or 2 additional hemagglutinin to the quadrivalent vaccines. And what they found was reactogenicity was similar in all groups. And when they added the H3 antigens, it prompted strain-specific responses and had no effect on the remaining parts of the vaccine, the H1 and the H3. And removal of the B Yamagata also reduced the responses, which is not surprising. This is a very complicated picture here, but this just shows you some of their data. And just the one that's in the box there shows that with 2 H3s, you get increased responses over a single H3 to the new component without affecting, in the bottom part of that graph, without affecting the H1 or the B components. So we have a number of different types of influenza vaccine available in the market. So we have recombinant protein, so the Sanofi recombinant hemagglutinin vaccine. We have live attenuated vaccines from AstraZeneca. We have a number of split inactivated vaccines with and without adjuvant. We're still waiting for mRNA to come up, and nobody has really developed anything remotely useful in the synthetic peptide area or virus-like particles, but these areas are in development. So looking at what's going to happen over the next few years, I was asked to estimate what was going to happen in 3 years' time, from 2022 to 2025. Really nothing's happened in terms of changes in the shared market of these different vaccines. We still have the egg standard dose vaccine dominating the global sales, with some sales of the high dose, the Sanofi product, and some sales of the MF59 egg adjuvanted product, and the live attenuated vaccine is still made in eggs, but there has been some movement in cell-derived vaccines over the years, and not much change in recombinant proteins. What's going to happen in the next 5 years? mRNA will definitely probably take a place here, but cell-adjuvanted vaccines will come into play, and eggs will start to reduce slowly, I think. So in terms of use of the seasonal influenza vaccine, there is quite a global use of that vaccine. It does vary from country to country. Some of the bigger countries, such as India and China and Indonesia, use very little vaccine. This graph is not exactly accurate because New Zealand used vaccine, and so does Japan, which they're coloured that they didn't respond to this survey. But only 8% of low-income countries and only 30% of low and middle-income countries use any vaccine for older adults, compared to 89% of high-income countries. And there is spare capacity out there. Currently, there's an estimated capacity of 1.53 billion doses of seasonal influenza vaccine. This was estimated in 2023. Actually, we only make about half that amount currently, so there's plenty of capacity, but it's only made in a certain number of countries around the world, Korea being one that has its own vaccine manufacturing. So the Holy Grail is a truly universal vaccine where we can cover everything. Currently, we can cover these. So we're struggling a little bit there. We can't cover all the influenza A's, we can't cover all the influenza A's and all the influenza B's, and we can't even cover the different variants that we see across a single subtype, such as H1 shown in the graphic there. So we were looking at this back in 2022, trying to look at things that might be effective against these different components here. So we came up with this graphic, and just to update this a little bit. So the green is what we'd already achieved, the yellow is what we thought we might be achieving in the next short while, and then red was still to be achieved. And that probably still is pretty much the case today. So if we look at what's changed in these proven effectiveness, we've got mRNA and we've got self-amplifying mRNA now. And for those of you who have heard Florian Kramer speak in recent years, he's a big advocate for adding neuraminidase to the vaccines. and hoping that that will have a positive effect on seasonal as well as pandemic vaccines. So when we're looking for enhanced longevity or duration of vaccines, we really don't have anything, I don't think, currently available. mRNA is unlikely to give us that increased duration, and this priming and boost might do something, but really hasn't been shown to be economically viable. Again, what will be effective against the H1 and the H3 influenza A's, if we add that recombinant neuraminidase, that might do something. Many of these other ones marked there have been tried and failed, such as the M2E has never come to commercial operation. In terms of effective against the group 1 or group 2, again, we're limited there. Maybe mRNA might be able to do that. And conserved elements, so instead of looking at the hemagglutinin or the neuraminidase, one of the more conserved proteins, the nuclear protein, has been targeted for some developmental work. And universal vaccines against all influenza A viruses, mRNA can do that, as has been shown in animal models, and I'll show you an example of that in a minute. And again, conserved elements might be of some use there, as they are against influenza A and B viruses. So a lot of work to do. mRNA is certainly the golden child of the moment, and it's many reasons for doing that. This is a slide taken from the Moderna website here, showing you what they're actually doing in terms of the space for respiratory vaccines. And you can see here that influenza vaccine and flu and COVID vaccines and pandemic flu are all on their radars, and they have completed phase 3 trials for flu and the flu plus COVID vaccines. And they've announced some of those results, and they look quite promising. They did a comparison to the standard dose seasonal influenza vaccine and showed a relative vaccine efficacy, so an increase compared to the standard of 26% in this 50-plus age group. And they also did an analysis on the 65-year age group, and it's compared to quite favourably to the high-dose Sanofi standard vaccine, which was an increased relative efficacy of 24.2%. And if you look at where that sits compared to the over 65s in the Moderna study, they had a relative VE of 27. So all very similar, but really no major improvement over the Sanofi high-dose vaccine. And the trouble with the current mRNA vaccines is the reactogenicity, especially in the younger age groups. So this is just on the left-hand panels there showing different responses in the standard. To the left, these are duplex type plots here. So we have a standard and then the mRNA. You can see a lot of those second blocks go above the 50% in the greater than 18-year-olds or the 18 to 64-year-olds, but much less reactogenicity in the over 65s, which I guess is a good target group for mRNA vaccines. And immunogenicity showed good responses using microneutralization against all components in their mRNA vaccine. Oops. Okay, and Pfizer have done the same, and their vaccine looks pretty good. Their combination influenza and COVID vaccine, again, this is just top-line results, and they showed good responses and safety and tolerability similar to single-dose mRNA vaccines, and the responses were similar for the influenza alone or the COVID-19 alone vaccines given at the same visit. What about the Holy Grail, putting 20 or more hemagglutinins into a single vaccine? Can that work? So this is work that's been done in mice only. So this is a study where they put all of the different hemagglutinins in, including the seasonal influenza A and influenza B components there, and you can see in those top graphs there, they're the responses to the different hemagglutinins, and you can see across the board they respond very well. And I think it's quite a promising technology, but whether this will work as well in humans. And the question really is, do we need something like this? When we look at what's circulating globally, I just pulled down all of the sequences here that have been on GizAid, since the 1st of January up until a week or so ago. And these are the hemagglutinin genes which have been sequenced. So you can see there that out of those 18 different viruses with different subtypes, only a few dominate there, and by far the greatest number is H5N1, so making up 88% of all of the viruses which have been sequenced this year. So maybe we don't need a 20-valent, maybe a 1 or 2 or 3-valent pandemic vaccine would be enough. And so we already have many of these vaccines available. So WHO, as part of its role, is it makes these vaccines by reverse genetics, and these are available as seed viruses for companies to rapidly produce vaccines if required, using all of those different platforms that I showed you earlier. So there's 3 vaccines to the current H5N1, this 2344B, so-called clade, the same one that's circulating globally and is infecting dairy cows in the US. And we're also making 2 new ones for viruses which are circulating in India and Bangladesh. That's the A Victoria type viruses, and also the viruses which have been infecting humans in Cambodia recently, another one under development. So what else is happening? So this is a different approach. I mentioned the nuclear protein approach. So this is a group in France, in Lyon, that are working on this nanoparticle type of approach here, where they self-assemble nuclear proteins, so a very conserved protein from influenza, anywhere between 98 and 100% homology between the influenza A strains, and even some homology between the influenza B strains. So these self-assemble into this nice little particle and have been shown to be quite immunogenic, so they've published on this. I don't think this could stand alone as a vaccine, but maybe in combination with the standard seasonal influenza vaccine may give some improvement and some higher vaccine effectiveness, and they're doing these sorts of studies now. Influenza B, there's some innovative ideas looking at influenza B as well. This is the Mount Sinai group, and they mess around with the hemagglutinin and take away some of the dominant regions with the influenza B virus to try and target some of the subdominant regions to improve cross-protection. And they do this by making reverse genetics and then rescuing those viruses and making vaccines in the conventional way. And they can show that they're immunogenic. However, they did need adjuvants to get improved responses. What about a different approach? So this is a different approach. This is a non-vaccine approach. And this is something which looks quite exciting. This is a group called Cidara, and they've developed this innovative type of molecule. This is using zanamivir. So zanamivir is a neuraminidase inhibitor, a quite effective drug against influenza. It inhibits all influenza types, influenza A and all influenza Bs. And it has these zanamivir dimers coupled to an FC component. So the FC component of the immunoglobulin, if you treat it right, if you make some changes there, it can help prolong the half-life of these molecules. And so that appears to be the rationale why they've chosen this. And it's given by a single subcutaneous injection. You only need a single dose per influenza season. Has a half-life of 50 days, which is pretty amazing. And to date, no anti-drug antibodies have been detected. But people have only received 1 or 2 doses so far. And you can store it at 2 to 8 degrees, and it's stable for at least 2 years. But what we don't know is the cost. So they've done some trials. They've done a 2B trial where they did a dose-ranging study, and they showed between 76 and 58% protection over a 24-week period compared to placebo. So they're now doing a phase 3 trial with 6,000 participants with one dose, 450 milligrams subcutaneous dose in adults and adolescents. And they're also going to be looking at immunocompromised patients, so that's interesting. So we have also been involved in a project with a small chemical entity produced by OzBio. And this is also based on zanamivir, as it turns out. So zanamivir to target the drug to the neuraminidase on the influenza surface. And our drug has also a secondary activity against the hemagglutinin. It produces an acidic environment which unravels the hemagglutinin as if it was being uncovered within a cell. So we've done a little bit of work on here, and we've shown that it's 1,000 times more effective in vitro compared to zanamivir. It doesn't do that by acting through being a super-neuraminidase. It has the same neuraminidase activity as zanamivir. It works against seasonal viruses, works against swine and influenza viruses in vitro, also works in mouse models here. So this is one of our lead candidates here, and you can see here that given up to 25 days before infection, that this drug is still effective at preventing weight loss and death in mice. So in summary, influenza vaccines have been produced for nearly 80 years. Currently, by far, the majority of them still made in embryonated hen's eggs, as they were back in the 1950s. Some advantages have been made. We've got manufacturing improvements. We now have cell culture-based vaccines. We have recombinant vaccines licensed. We have high-dose or adjuvanted vaccines for the elderly. And live attenuated vaccines are still a very good vaccine for children, but unable to deliver a highly effective virus against circulating virus. We can't broaden the immune response to cover emerging viruses, and we've failed to increase the duration of protection, and vaccines still require updates every year. So new technologies may help out. We've still got a long way to go to have the ideal vaccine for all ages, for seasonal and both pandemic viruses. And there is competition out there from long-lasting antivirals, such as the Cidara CD388 FC-linked zanamivir. So just thank you all for listening and suffering through that initial problem, and here's all of the people that do the work. Thank you.